my independent research project is called A Deeper Look into Antidepressant Dependency, and it's an investigation on the hepatic metabolic processes behind enzyme cytochrome P450 2C19. You will see what that means in like a second. Okay, so introducing the worldview, just a big introduction to generally what I'm going to be talking about, because I can, like, a lot of people here, their research project has been talking about salinity, like the ocean, and climate change, and all those things are very clearly, especially here at TAMUG, they all affect us, and we all think about it every time, like even ASEC, it's like, it's right there. But many of you may be wondering, why would research on antidepressants matter to you or me, even if I don't take the medicine? So I will show you. So, does anyone have a guess for what this is? Protein?
Protein. It's a protein.
Yes, it is protein. So it is an enzyme from cytochrome P450. And so cytochrome P450 is a huge superfamily of enzymes found in all kingdoms of life. I thought it was just like a human thing, but they're even in viruses, which is really cool. And so there are over 300,000 distinct CYP proteins in mammals alone. And in humans, these proteins are found in the inner membrane of the mitochondria or the endoplasmic reticulum of cells, so that means it's basically everywhere, but they are primarily concentrated in the liver, which is why I'm studying the hepatic metabolism. Okay, and so to help you understand further about cytochrome P450, the CYPs and P450s, that's like what they call both the genes and the enzymes. And so it can get a little confusing because it's like, oh, this is CYP this, or CYP that, and it could be talking about the enzyme or the gene, but you just specify whenever you're talking about it. And so, as you guys guessed, it is a protein, it's an enzyme. All those ribbons is the enzyme part. And in the middle, along that like cysteine chain and everything, that's the enzyme active site. And we all know that about like enzymes, that the main part is the active site where things happen. And so, yeah, this, as I said, cytochrome P450 is in every single one of us. And they have an incredible impact in how we function every single day. It goes from like, synthesizing hormones from estrogen and testosterone, to metabolizing vitamin D, breaking down cholesterol, and most importantly, breaking down possible toxins in the liver, which includes alcohol and every single medicine that you take. So they oxidize steroids, fatty acids, synthesize hormones, and metabolize thousands of modern drugs. So from antidepressants, which I'll be talking about, to like your ibuprofen that you took yesterday. And in the model, you'll see where the arrow is pointing to, the little white bar thing. It's just representing the drug molecule that will be at the active site. And this one is modeled by CAMP4, but it can be anything. It will, in my situation, it will be this antidepressant. And it is meeting at the active site of this CYP450 enzyme, and it's being oxidized. Okay. So, oh, yeah, going back to that, it metabolizes thousands of modern drugs like these. These are the top 16 prescribed drugs of 2020 in the United States. And as you can see, there are, like, I highlighted the different categories of them, and they're all common American problems, like cardiovascular and heart disease problems, diabetic problems, and like hyperlipidemic, which is like high fat in your tissues and everything. But what surprised me, actually, was that 2 antidepressants are in this top 16 list. I was not expecting that. I really thought it would be a majority cardiovascular and other things like that. But both sertraline and citalopram are in this top 16 prescribed drugs. And so it affects so many people, way more than we thought, way more than you would have expected. And it's more common than you think. It was more common than I thought. Fuck. Okay. Through the study of antidepressants and the enzymes that metabolize them, we can learn so much about the way our bodies handle medicine. like if you just look at the amount of prescriptions in 1 year, 38 million prescriptions for just sertraline, and then escitalopram is over 30 million. So I will be studying escitalopram. It's also known under the brand name Lexapro. That is the structure of it, and also like the Molview structure of it. This antidepressant is used to treat major depressive disorder and generalized anxiety disorder, so both of these. And it exists under the subcategory of antidepressants. There are many different kinds of antidepressants, but the most prescribed ones are SSRIs, which is selective serotonin reuptake inhibitors. A lot of words, but we will get to it. So this is going to be the substrate or the drug that I will be studying in this. So let's go more into selective serotonin reuptake inhibitors. I wasn't familiar with this, but it's actually very fascinating. So what some of you may not have known about major depressive disorder or generalized anxiety disorder is, and a lot of other disorders, is that their symptoms are created from the chemical level. They begin with a serotonin imbalance in the neural system, and it's caused by a misuse of the reuptake mechanism. It's kind of like whenever you have neurotransmitters in your coming across the synapse, and there's too much of it, the presynaptic nerve will be like, oh, let me take these back up. Let me take these neurotransmitters back. There's too much of them, and we'll just take them back up. But people with MDD and GAD have a misuse of this symptom, and so on the schematic that I made, you can see like in the top section, all those like little yellow circles are the neurotransmitters of serotonin, and the blue arrow is all of those the neurotransmitters being reabsorbed into the presynaptic nerve ending, even though all of those serotonin receptor sites on the other end are empty. And so people can have serotonin have serotonin sent out into their body, but it isn't correctly absorbed. And so SSRIs, once you take them, it inhibits this reuptake mechanism, and it's the green little like protein, and it blocks reabsorption, and it allows it to cross the synapse and head to the serotonin receptor site across like the neural network. And so by adjusting the antidepressant, it stops the reuptake and allows people to have serotonin. And so why this really fascinated me was because obviously that diagram was very fascinating in the first place, to see that something as physically presentable as a disorder, like major depressive disorder or generalized anxiety disorder, can come back all the way to just neurotransmitters not being sent across the synapse, which is, I thought that was really cool. But I looked further into this, and of course, people with emotional disorders are incredibly susceptible to other things, but there are many, many studies on confirmed comorbidity of alcohol use disorders and MDD and GAD. And yeah, so many studies have been done, and it's very, very comorbid. And so I question this because going back to that enzyme that we talked about, that enzyme lives in the liver. And we talked about what happens in the brain and then nerves, but going before that medicine heads to your neural network, it has to be filtered in your liver, like any other pharmaceutical. And what else goes through your liver? Alcohol. For people with MDD and GAD that are much more likely to exhibit an alcohol use disorder, they have a large amount of alcohol passing through their liver, which can change if you go back to here. Excessive alcohol use can lead to a temporary but significant change in the liver microsome's pH. And with what we know about enzymes, I forgot who talked about, I think it was just Anushree, talking about denaturing enzymes, and I don't know if it was you, somebody else. There are specific parameters. You. Okay, there are specific parameters for enzymes and how they work, and any change in this pH may change how well that enzyme can metabolize your medicine. And so I will be specifically studying enzyme CYP2C19. So that goes all the way back to the title with that bunch of numbers and letters. And this is one of the more major and prevalently talked about enzymes in the cytochrome P450. And it's the main enzyme it's the primary enzyme that metabolizes acetylopram. Okay, finally to my hypothesis. Okay, if all possible toxins are filtered into the liver, how do major pH changers like alcohol alter the productivity of CYP2C19 and therefore the effectiveness of acetylopram? A lot of words, but just like I said, if everything goes through the liver, how do things like alcohol change the liver microbiome and its parameters in pH, and how does that change the effectiveness of one's enzyme, and therefore how does that change how well your medicine works? And even further, just for the future, I wanted to wonder, can a reoccurring addiction to alcohol change hepatic pH? One so unideal for CYP2C19 that it becomes completely denatured. Okay, and alcoholism effect on the liver microbiome. In the study, I will use acetaldehyde, and you may be wondering why I'm not using specifically ethanol or just like straight up alcohol. It's because acetaldehyde is created when P450 liver enzymes, different than this one, but they do the same, different things. They break down alcohol, and it is only active after a person has consumed a large amount of alcohol, which is indicative of alcohol addiction or misuse. And that's the breakdown of pure ethyl alcohol into acetaldehyde, which later is broken down into acetate. But I will be using acetaldehyde in this experiment. So my method. This sounds really complicated, but I got really lucky because a company called Xenotech has already come out with a assay kit for the top 15 most prevalent enzymes in P450. And so I got really lucky that I just get to follow along this. But when I was doing my research, they are impossibly expensive. I don't know if you can see, but for 81 vials of human liver microsome, it's $10,000. And so that's crazy. But yes, I will still be following this just because this is a proposal and I'm not putting it into reality just yet. And I don't know if I will, especially with that price tag, but it's okay. So the following experiment will be done in vitro, which is just like in test tubes, not in a person. And I do have to acknowledge the difficulty in both expenses and scientific paperwork regarding using human liver microsomes, but this is all theoretical for now and just proposals, so I'll follow through with the methodology. So, that top section is a screenshot from the assay kit's description, and I separated them into just the different sections of what they are. Okay. So I don't know if you remember, but when we went on our salinity trip, boat trip, we took just samples of water and put them in those little tubes. I am going to be using those same exact tubes in this situation. Those liver microsomes, and they're if you are a MARB person or anything, they almost look like mini versions of sediment cores. They're tiny little cylindrical tubes of someone's liver. And so they're thawed at around 4 degrees Celsius, and then they're brought up to 37 degrees Celsius to mimic the temperature of being inside of a person. So also inside this tube is the substrate, which will be acetylopram, this medicine, and then it is dissolved into a little amount amount of organic solvent, so it, like a buffer, but not too much because it actually like hurts the substrate. And then the last three components of like the glucose phosphate and NADP to like create the energy are just thrown in there. And so these are done in several different trials in increasing amounts of acetaldehyde. So everything, the control is all the basal conditions described in conditions, just the microsome, the substrate, and then the energy components. And then the trials are increasing amounts of acetaldehyde proportional to the amount that would be experienced in the liver. So the extraction of the results. Oh, also, I got the chance to look at someone else's experiment on alcoholism in the liver in a different way, and these are their experiments in a petri dish. And you can see like the different ways that things look whenever alcohol is exposed to a liver microsome. And mine won't look exactly like that, but they do look cool. So the extraction of the results, since inside of that tubule is the enzyme, the substrate, and the components. And what's supposed to happen at ideal conditions is that the enzyme metabolizes the substrate, and then the little protein, all the way back to that picture where there's that little green thing that blocks reuptake, it creates that protein. But what I'm hoping to see is that acetaldehyde stops or denatures the enzyme in some sort of way, and we do not see that protein created. And the way that you see that is that you put those tubules into like the little smaller tubes, and you put them into a benchtop centrifuge, and the protein is pelleted after several rounds of centrifuging if there is a protein. And so the resulting protein is studied for mass to see like how much there is, if there's a significant amount, and then the supernatant in the tube is assessed for like any leftover citalopram substrate. And so that makes no sense, but interpreting what you see will help us understand. So to help recap what I just said is, remember my hypothesis was if acetaldehyde is added to this control, this condition, it will inhibit the metabolism of the substrate by CYP to C19. And so when you take out that protein and you study the supernatant, if there is no protein created, it will show that CYP to C19 did not work and was denatured by acetaldehyde if there's no resulting protein. But if it's the opposite, that there is a protein and then there's no more substrate left in the supernatant, it'll disprove my hypothesis and say that oh, it still worked. Even like adding acetaldehyde, adding alcohol didn't change it may have changed the pH, but the pH didn't matter in denaturing the enzyme itself. So we'll just have to see if I get to do something like this, what it will look like, because I don't know yet. Nobody has done something really kind of like this. And so the impact, the implications. I was wondering what the implications were because I was like, oh, addiction, blah, 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 blah. It obviously has a very deep implication, especially going back to how many people take antidepressants and how many people also experience comorbid alcohol use disorder. Like, the impact of the study reflects so much more than just one single enzyme in your liver and how it works under different pH. It actually goes further. I know that you probably understand that when somebody takes a certain dosage of medicine and then they're like, oh, this isn't working for me anymore, and so they take more, and then they take more, and they take more. That's tolerance to a pharmaceutical drug. And it just gets worse and worse because it can have a greater impact on your body the greater amount of prescription that you have. But in this situation, somebody could be taking an antidepressant like citalopram and have an alcohol use disorder. And in the case that my hypothesis is proven, if they may keep taking higher and higher doses of that antidepressant, not knowing that it's the alcohol that is denaturing the enzyme that metabolizes their medicine. It's not the medicine that isn't working, it's the alcoholism. And that's a hard thing to realize, especially whenever you don't understand. I don't know who goes back to the enzyme that metabolizes their ibuprofen or their allergy medicine. And this is a really important study because a lot of people don't look back on that, and it's very, very common. And in the case that my hypothesis is proven, it could be very impactful as well. These are my references. There's a bunch. Thank you.
I don't have a question, but I do have a comment.
Okay.
I think a really good way to get funding for your experiment is to appeal to emotion.
So true. Okay.
Yeah.
Anyone have a?
I have a few.
I will. Okay.
Oh, me?
Yeah.
Okay, so I'm not 100% well versed in this, but I was gonna ask, when a doctor usually prescribes an antidepressant,
Yes.
don't they try, like, and they know the patient is dealing with alcohol, like, addiction, don't they try to make sure the person is going to get one therapy and to try, like, is attempting to be sober or is already sober?
Okay. Yeah. I think, of course, in an ideal situation.
Like, obviously the person's gonna go by
Yes.
because they are 21.
Yes. But I know we all know that any healthcare system is not going to be perfect, and I know you know that too. But of course, in an ideal situation, somebody would be taking care of their alcoholism before they take antidepressants or while they take them under careful monitoring. But a lot of times, especially with alcoholism, people like to keep that private, especially like, not even going into that, but people do like to keep that private, and people don't tell their doctors. And sometimes doctors don't even ask. If you look at like acetylopram or any other antidepressant on like PubChem, you'll have to scroll all the way down before you see like, hey, don't drink alcohol with this, or don't do this with that. And so it's not really widely talked about, but it's kind of understood with doctors, but they don't talk about it sometimes. So, okay.
Okay, a few questions, and it's okay, because this is a really complex topic. So you mentioned that the protein gets denatured with acetaldehyde. Do you have sources for that? I guess it just acts as an inhibitor.
I guess that's what what I'm finding out, to see if acetaldehyde is enough of a pH changer or enough of a denaturer to change the enzyme's productivity.
So yeah, so inhibitors would lower the activity of the
Yes.
enzyme, and there are different kinds of inhibitors. So basically what inhibitors do is they would either hit on the active site or they would bind on the other parts of the enzyme, but not necessarily like nature.
Yeah.
So I would rephrase it,
Okay.
just simply say it can act as an inhibitor.
Okay.
Yeah.
I think there's on the assay kit they have like a bunch of different methods that they do to test like a new pharmaceutical for that superfamily, and the one that I'm doing is like changing metabolism, and so like what I said, in creating the protein, and I think it's that's definitely the crux of where I am in chemistry right now and my understanding of this, because I try to understand. Oh, sorry. I tried to understand like how the medicine interacts with the enzyme itself, but that goes into like different structures that I don't. So yeah, but hopefully I'll be able to learn like how that works, and whether or not it's like a specific kind of inhibitor.
There's another question for you.
Oh, hi.
But the cytochrome that you're referring to, is it like, I'm kind of confused by how you mean it participates in metabolism, because are you referring to like the cytochrome in your mitochondria that is in the ETC? Or is this like another cytochrome that like actively like breaks down and like processes
Yeah.
biomolecules that we want to consume?
Like, I had to look this up too, because I was like, why use the word cytochrome? I don't even know. Like, but I do know that it does participate in, I did mention like that it like participates in a ton of different things, but I don't even know the extent. And I think it would probably mean something different. I'm assuming that cytochrome in this context means something along like the lines of like the superfamily rather than like the structure and what these.
And there are different kinds
So this is in vitro experiment. I imagine, can you find some substitute to reduce the, like we have already discussed, do you, instead of using human
Yes.
sample, do you have find out if it's possible to use any substitute?
Yes. Okay, so we talked about this whenever I came to lab because we were concerned about the budget of how much it would be. But since we know that P450 is in every kingdom, it would be very plausible to use liver microsomes of different animals. And I'm assuming pigs. I didn't look at it that much since the time but I'm pretty sure I got to see some things, and it's, I forgot what kind, but it's pigs. Yeah, so that might cut down the cost a lot. So yeah.
That's a very thorough. I really like how you introduce your hypothesis and how you present your hypothesis, if it's proven or it's a great job.
Yeah.
Thank you.